Hello, my name is Melina Liu. Today my poster is on my master's thesis on a metadata review of archaeological dental calculus studies published between 2000 and 2020. So dental calculus is calcified dental plaque, which has been used as an indicator of diet and other behaviors of humans in archaeological studies. So from calculus, one can extract microremains that are deposited through various processes, such as phytoliths and starch grains, which are remains from plant foods consumed during an individual's life. So for my review, I based my work off of Lievers 1999, who did a brief survey of calculus reviews to discuss how calculus is formed and what factors may influence this during a person's lifetime. So my review expands upon this by examining how calculus has been used from 2000 to 2020 as indicators of diet and other behaviors, but with major emphasis on microremain extraction due to various technological and methodological changes that have taken place in archaeology since Lievers' publication. So my review intends to answer the following questions. Number one, what sample sizes are employed in research? 2, what specific microremains are favored for extraction? 3 and 4, what regions and time periods are favored? And 5 and 6, where in the teeth and which teeth are used for extraction? So my methods involved using Google Scholar and the ISI Web of Knowledge for calculus papers published between 2000 and 2020, recording and analyzing 12 variables using an R-Shiny application, though this is an abbreviated version with 6 variables. Exclusion criteria varied. For example, reviews may be excluded from sample count analysis because they didn't actually conduct analysis of calculus or extraction. So importantly for this review, I operationalized all the potential time ranges by assigning a number to each millennium using the BC AD time system to lessen the variation between time ranges. So for example, -1 means 1000 to 0 BC, while 1 means 1 AD to 1000 AD. So for my results, according to figure 1, almost 90% of studies had sample sizes of less than 50, which could be due to potential budget limitations or lack of access to large sample sizes or archaeological sites, though it should be noted some studies will emphasize unique individuals. For example, Gismondi et al. 2020 study on the calculus and diet of an ancient Roman woman with the earliest documented case of celiac disease. According to figure 2 over here, 68% of studies included phytolith and or starch grains, though it should be noted around 40% of studies extracted more than 3 types, meaning they may be taking anything possible from the calculus because standard chemical extraction may yield many different microscopic remains, while more specific methods such as DNA analysis generally yields only 1 or 2 types. Figure 3 over here shows Europe was the most commonly studied region at 49%, showing a very sharp rise in publications in 2015, which again could also be due to site and sample accessibility. Figure 4 shows that 77% of all studies took place between 3000 BC to 2000 AD, so -3 to 2, which could be attributed to deterioration of the human remains over time through taphonomic agents. So the older the samples are, the less potential there is for extraction. Finally, figures 5 and 6 down here. Figure 6 shows about 44% of all studies fail to disclose the type of teeth sampled from, such as the incisors, while in figure 5, 69% did not disclose where samples on the teeth were taken from, such as above or below the gum line, so subgingival versus supra. So this may be because most studies do not have the time to individually sample teeth, though a study by Dudgeon and Trump 2014 shows that there are potentially differences in what microremains may be found on different teeth. So for example, they found potential sugarcane phytoliths on the back teeth, which requires more mastication, while very few on the front teeth because the front teeth are not as capable of intense chewing. So overall, my conclusions are that we must expand dental calculus studies to further enhance what can be extracted and learned. So therefore, researchers should attempt to studies on more regions in the world, such as Africa, more time periods older than 3000 BC, as well as engaging in more specific reporting and sampling of what teeth are sampled from and where on those teeth. Increased specificity will enhance how much we can learn about the diets and behaviors of ancient peoples by allowing researchers to further contextualize how these microremains may have come about using, for example, time periods and regions from the first point of consumption in the human body. Thank you.